You should remember that there are specific types of white blood cells called lymphocytes that produce antibodies. These antibodies target specific antigens usually found on the surface of pathogens. You should also remember that there are tumour cells, and unlike white blood cells, these cells do not produce antibodies, but they do have the ability to divide rapidly and make clones of themselves. Some scientists have developed a way of fusing these two types of cells so that you get the combined property of making antibodies that are specific to an antigen and being able to clone these cells and have lots of them. This means you can produce lots of a specific type of antibody. They call the cells that they make in this fusion hybridoma cells. And the antibodies that are produced are called monoclonal antibodies. Let's see how they are produced. To produce monoclonal antibodies, scientists have injected mice with antigens. These antigens will stimulate the B lymphocytes within the mouse to produce antibodies that are specific to that antigen. The scientists then take tumour cells that are able to divide rapidly and fuse these two types of cells. The fusion produces hybridoma cells, and these hybridoma cells have particular properties. They are both able to produce specific antibodies and divide rapidly. So the cells can then be cloned to produce a hybridoma cell culture. The hybridoma cells will then produce lots of the same antibodies. These antibodies can then be collected and purified. They are called monoclonal antibodies, as the antibodies are produced from a single clone of cells. More recently, scientists have combined the mice cells with human cells as well to produce monoclonal antibodies that are less likely to be rejected when used in humans. Antigens are protein molecules that are often found on the surface of all cells, although free protein molecules can also act as antigens. The monoclonal antibodies produced from a single clone of cells are specific to bind to one specific antigen. This antigen might be found on a specific type of cell in the body, or a particular chemical. Because the monoclonal antibodies target only specific antigens, they can be used in a number of ways. This includes pregnancy tests, disease diagnosis, measuring and monitoring health conditions, treating diseases, and also advancing scientific research. The monoclonal antibodies in a pregnancy test are sensitive to the presence of HCG, human chorionic gonadotropin, which is a hormone made in the early stages of pregnancy. Tiny amounts of the hormone are passed out of the body in urine. They then pass through the pregnancy test strip. They first pass through the reaction zone. In the reaction zone, there are mobile monoclonal antibodies that specifically bind to the hormone HCG. These antibodies are then able to move to other areas within the pregnancy test and have a coloured dye attached to them. As these mobile monoclonal antibodies move up the pregnancy test strip, they reach the results window. At the results window, more immobilized monoclonal antibodies specifically bind to HCG. The HCG has already bound to monoclonal antibodies in the reaction zone. So you now have two monoclonal antibodies binding to HCG. This allows the dye from the first monoclonal antibody to be released and seen in the results window. If there is no HCG present, so the person is not pregnant, then no binding will occur in either the reaction zone or the results window, and therefore no dye will be released. The final window is the control window. Here, there are more monoclonal antibodies. These antibodies are immobilized, and they are not specific for HCG anymore, but for the original monoclonal antibodies found in the reaction zone. Binding of the immobilized antibodies to the original reaction zone antibodies will cause release of the dye, and therefore a coloured strip will appear in the control window. This control window should have a coloured strip for whether a person taking a pregnancy test is pregnant or isn't pregnant as a control. It is the results window you then have to look at to see whether it is a positive or negative result. Monoclonal antibodies can also be used for the diagnosis of disease and measurement and monitoring of different health conditions. In the diagnosis of disease, monoclonal antibodies bind to specific antigens found on pathogens. All the antigens could be found on blood clots or even cancer cells. The monoclonal antibodies can also carry markers that make it easy for doctors to see where they have built up. This allows doctors to detect problems before they seriously affect a patient's health. For example, a blood test can be used to diagnose prostate cancer using monoclonal antibodies. To measure and monitor conditions, monoclonal antibodies are used in hospitals and laboratories. And just like in a pregnancy test, can attach to hormones or other chemicals in the blood to monitor the different levels as indicators of health. This is very useful for blood screening for the progression of diseases like HIV, or also the detection of illegal substances that may be used by athletes. Scientific research has also been advanced by monoclonal antibodies. Research scientists use monoclonal antibodies to locate and identify specific molecules in a cell or tissue. Scientists produce monoclonal antibodies linked to a molecule of fluorescent dye. And when the monoclonal antibody binds to the desired molecules, scientists can see where this has happened by observing the build-up of fluorescence. There have been recent advances in the use of monoclonal antibodies in cancer treatment. Cancers are often hard to treat because they're hard for medicines to access directly, whereas monoclonal antibodies could be used to target specific cancer cells and not harm healthy cells of patients. Currently, there are 3 different ways of using monoclonal antibodies to treat cancers. Each has produced some cancer treatments that are being used, and many more are at the clinical trials phase. The first of these is the direct use of monoclonal antibodies to trigger the immune system to recognize, attack, and destroy cancer cells. The second is the use of monoclonal antibodies to block receptors on the surface of cancer cells and stop them from growing or dividing. The second is to use monoclonal antibodies to carry toxic drugs or radioactive substances for radiotherapy, or chemicals that stop the cell growing and dividing, to attack the cancer cells directly without harming other cells. The third is using monoclonal antibodies to block receptors on the surface of the cancer cells, and this will then stop the cancer cells from being able to grow or divide. Although monoclonal antibodies pose great advances for scientific research and disease treatment, there are also risks involved in their use. So the benefits of monoclonal antibodies is that they can be used to bind specifically to target sites and not affect healthy cells. And this specificity in binding also means many more diseases that couldn't be treated before may now be able to be treated. However, monoclonal antibodies are very expensive to develop and so far have produced much more side effects than were expected in their use. This is probably due to the use of mouse cells in hybridoma formation. If you enjoyed this video, make sure you give it a like. And if you have any questions, leave a comment below. Check out more of our content and remember to subscribe to our channel.